All right, thank you everybody for joining, everybody in the room, everybody virtually. We've got Gideon Lapidoth here. He is co-founder, CEO of Enzymit, which is a Fund 2 portfolio company of First Stars, doing some really exciting work in enzyme and synthetic bio work. I'll let him give a quick intro of himself and his team as well, but hopefully going to learn a bunch about what these guys are up to.
All right, perfect. Thanks so much, Teddy, and the First Stars team for arranging this event. So before I go into Enzymit's story and how it fits in the whole bio-revolution that we're going through right now, a little bit about my background and what got me to this point. I did my PhD in computational protein design at the Weizmann Institute. Before that, I had an undergrad in CS and biology. So when I did my PhD in computational design was still a relatively niche area. It doesn't have the wide recognition it has now with the success of AlphaFold and all the interest coming in from the big software companies like Microsoft, Google, Facebook. So we're definitely going through a very interesting, exciting time. So as I said, I finished my PhD, stayed on for a short postdoc, also at the Weizmann Institute, focusing on enzyme engineering, and really decided that the technology has reached a level of maturity where we can spin it out to a company. So I joined forces with Dror, my co-founder, who is also an alum of the Weizmann Institute. We met at the same lab, so a lot of know-how and proficiency in enzyme engineering. Dror graduated before I did, so he did a stint at a few other startups, mainly focusing on antibody design. And when I called him over, he was more than happy to join me, and this is what got Enzymit off the ground. So going back to where Enzymit fits in the whole ecosystem, essentially we want to make bioproduction the norm and not the exception, like it still is, unfortunately. And the way we're doing that is by building a new technology called cell-free production, which I'll talk about in a few slides, but essentially it just means taking away all the complexities and unnecessary parts and just keeping the bare essentials to enable biomanufacturing. And this, essentially what it means is it gives us cheaper, faster, and scalable bioproduction. So before I talk about more of the technology, a bit of a primer on what bioproduction is. So essentially bioproduction is any type of manufacturing that uses some sort of biological component to produce pretty much whatever. So it either could be DNA or enzymes, this funny looking Pac-Man here in the middle, or whole bacteria in cells, which is the way that bioproduction is currently mainly done. And our vision and mission is to make bioproduction not only environmentally friendly, which it is almost inherently, but show that it can be done cost effectively and compete head on with chemical manufacturing. So the number that I have up here, the $4 trillion number, was taken from a McKinsey report that was put up about 2 years ago, and it predicts that bioproduction could be a $4 trillion industry by the end of the decade. Unfortunately, when you take a snapshot of currently where we are, we can see that fermentation of chemical manufacturing is about an $86 billion industry versus the chemical production industry, which is currently a $4 trillion industry. And that's really what we're looking to change. So we want to take bioproduction and overtake chemical manufacturing. So take over that $4 trillion market. So I've talked a little bit about cell-free, I've talked about fermentation. So what's really the difference between the two? What exactly are we talking about? So fermentation is really the main workhorse of the biomanufacturing industry today. And essentially it's the same technology that we've been using for thousands of years, where we take bacteria or yeast and use their natural processes to convert sugar into some sort of desired product. So it could be ethanol is a prominent example of that. So you can think of a bacteria as kind of like a manufacturing plant, and the feedstock is sugar, and the plant does a lot of processing, and it produces your output, but it also uses some of that sugar as fuel, and it burns it up and releases carbon dioxide into the air. And what's really doing the interesting chemistry inside the bacteria are these small molecular machines called enzymes. And each enzyme can do a very specific chemical process. So a typical bacteria has, over 1,000 different types of enzymes, And honestly, a lot of those really interfere with what we're trying to produce. So we don't need all those interfering enzymes if we want to produce a single molecule. And this is really where cell-free has a huge advantage. So instead of using a bacteria, which is essentially a sac filled with thousands of different enzymes, that 99.9% of them are just getting in the way, we're just using the specific enzymes that we need in order to convert our input feedstock into the desired output. And one of the immediate advantages that I hope you can see from this slide is that enzymes don't need sugar as energy, so 100% of the sugar gets converted to 100% of your desired output. So beside that, there are a number of very obvious advantages for using cell-free, and the number one advantage is just a huge reduction in capital costs. So the 12,000x number is a comparison between how much output you would get from a 600,000 liter fermenter, which for comparison is about the size of a 6-story building. So you can get the same output from this huge fermenter from a 50-liter bioreactor filled with enzymes. So 50 liters is less than the size of the room that we're sitting in. So right there, you already have a huge reduction in cost by just needing less steel and less cement in the ground. After that, a huge pain point for fermentation processes is a scale-up. So a fermentation process that works on the laptop doesn't necessarily scale up to an industrial process. So keep in mind, you have to keep cells alive and happy and growing and multiplying, and that takes up a lot of energy and know-how. And the scale-up is not self-evident. On the contrary, with cell-free manufacturing, enzymes are just molecules. So the scale-up is almost linear. If you want to get 10 times the production, use 10 times the enzymes, or just run them for 10 times as long. As I mentioned before, yields are much better in a cell-free process, so you don't use up any of your input stock to keep the cells alive or you produce energy. You don't need that in a cell-free process. And lastly, you can also produce molecules that you just can't produce in fermentation. So an immediate example would be something like antibiotics. So you can't use bacteria to produce antibiotics because it's just toxic to the cell. It will kill the organism manufacturing whatever it is you're trying to manufacture. So I guess the follow-up question to all this promise would be, if cell-free is so great, then why isn't everybody using cell-free? And the answer is that the key component for cost-effective cell-free manufacturing are the enzymes, the actual machines that do the production. So there are 3 components that are important to make cell-free a reality. Number one, cost of enzyme manufacturing. So just like any other factory, there's a capital cost. So the cost of buying the machine, or in this case, buying the enzyme. So the cheaper you can make the enzyme, the less you have to spend on the process itself. The second component, which is really where Enzymit is shining right now, is the stability of the enzymes. So the longer you can run your enzyme for, the more bang for your buck. And this has really been a huge pain point for enzyme engineers over the past, I would say, 2 decades. And the technology we've developed, we've really shown a huge improvement in enzyme stability using Enzymit technology. And lastly is, how do you feed energy to your reactions? So as I said before, in fermentation, you use sugar. You can't, and we don't want to use sugar as a fuel for these chemical synthesis and cell-free reaction. So we've actually designed enzymes that can use other energy inputs that are not sugar and are very, very cheap to use to drive chemical synthesis in a cell-free environment. So if you have all 3 of these, you have a very, very cost-effective cell-free manufacturing process. In order to unlock these capabilities, Enzymit's platform technology is addressed to do exactly that. So we have cutting-edge computational design algorithms to design novel proteins. We have a high-throughput screening platform, so we can test billions of different enzymes in a single experiment. And we're using a lot of AI to both drive the design and also extract useful information from the experiment. So kind of to condense everything that I said in the previous slides, if you have very stable enzymes that can run for a long period of time at a low cost of manufacturing, in a very simple back-of-the-envelope calculation, you can show that 1 kilogram of enzyme that can produce 18 tons of product. If you do the math, you find out that the cost of the enzyme itself is just 1 penny for the cost of the product. So essentially the enzyme cost factor is non-consequential for whatever you're trying to produce. The real cost would be just the cost of the raw material. So describe a little bit of what we've been doing so far. Enzymit, when we started, we had the computational platform, and immediately we started talking to industry partners and saying, listen, we have enzyme design technology, and we want to, what does the market think? And there was a lot of interest from the market, and we did a lot of collaboration. So a lot of companies that don't have enzyme engineering capabilities want to use enzymes or are already using enzymes in their manufacturing process. So they came to us and asked us if we can help them design better enzymes, which we did. So we've worked pretty much across every industry you can think of, from fine chemicals to plastic manufacturing to the cultivated meat industry to food ingredient manufacturing, and all of this was done to kind of first allow us to test our technology in the real world, so to speak, and also to learn from the market where the real need is. So after going through this phase, what we're really focusing on now is we've just launched 3 programs, and we want to show by the end of the year that we can produce sufficient quantities for each of these molecules, and I'll explain in a bit what they are. But the key differentiation factor here to show that we can do it in a very cost competitive way. So there are a lot of numbers here. So there are a lot of numbers here, but really what I want you to see here is, so these 3 products are already products that are on the market. Jet fuel, the price that I have here is the actual a cost for petrojet, so not biojet. But for the 2 others, these are products that are currently produced in fermentation. And you can see that the cost for production is pretty high. And when we look at what we can get to, our aim is to get below $10 per kilogram. So from 5x to 10x reduction in cost.
And one quick question here, because these are all very different end products. I'm curious if you can walk us through a little bit of why these first 3 products and kind of where their overlap is between them and what capabilities they're helping you guys build on the way as well.
Perfect. So yeah, it's a great question. So I'll explain a little bit what the products are, and then I'll explain the kind of the underlying connection there. So cumulative oligosaccharides are a type of what's called an oligosaccharide. Essentially, if sucrose is a simple disaccharide, fructose is a monosaccharide. If you connect a few of them together, you get a small chain. So that's the oligo word. And these are very specific prebiotics that are only produced in breast milk. So there's a lot of interest in the industry now to add these to infant formula. So these are sugars only found in breast milk, but they're very high to very expensive to manufacture, and that's why you only see them now in certain premium brands. Our goal is to get them to a cost where essentially other infant formulas, for those who have small children at home, use a different type of prebiotic, which is cheaper to make, a prebiotic called GOS. But those are produced also enzymatically at a very low cost. So for this program, our target is to show that we can do these HMOs at a cost which essentially opens up the full market. So it's not going to go just to the high-end premium infant formulas, but across the board. Hyaluronic acid, again, is another type of polysaccharide, different use cases. So it's used a lot for pharma and cosmetics, so injectables, lubricants. Another interesting use case where the raw material is just sugar, but because the manufacturing is so expensive, you get these enormously high costs per kg of product. And lastly, jet fuel, essentially what it is, you're converting. So currently, jet fuel, or essentially almost any other petrol fuel, is taking hydrocarbons from the ground, but you can also do this from a renewable resource by taking oils from vegetables and doing an enzymatic conversion to take these oils and take them and make them into hydrocarbons. So 3 different, apparently seemingly different products, but the underlying connection here is that these are all hydrocarbons. So essentially, we're building a know-how and capabilities in a hydrocarbon synthesis. And this is why you can run the gamut between seemingly very different markets and applications, from fuels to sugars. This could also be polymers for plastics, for example. I mean, there's really no scarcity in different applications. So this is kind of the very quick rundown of what Enzymit and where our current direction is. So if you guys have any questions, or Teddy, do you have anything you want to ask? or kind of?
One of my questions on this piece of it is, as you guys are thinking about, these 3 early programs, and as they kind of start to, emerge commercially, what do you think, other folks are going after these things as well, and there's a lot of different approaches. What do you think the kind of the market for these HMOs looks like if you guys get to the scale you need to get to and get to the cost level you need to get to? Do you think that entirely replaces GOS? I mean, if you guys nail the biojet fuel piece, what do you think? A little bit just in terms of the customer adoption piece. You guys make this stuff, and then folks have to buy it, integrate it.
Right. So that's again why I think cost. If my own personal opinion is that cost is the #1 driver. I mean, if you want to really change behavior, you either add a financial penalty or you reduce the cost, and that's how you get market adoption. So, for example, with HMOs, if we get to a price parity with the current prebiotics used in the market, and we know for a fact that there's a ton of clinical research showing that HMOs, this is what is actually producing breast milk. There's really no kind of debate here. You can have a better product at the same cost. I mean, the adoption here is almost evident. Hyaluronic acid, there's also a component of quality. So one of the challenges for fermentation, for example, because it's a pharma-grade product, the process of making hyaluronic acid fermentation is very, very expensive and very complex. Because imagine that a bacteria that is doing fermentation produces a lot of molecules alongside your target molecule. And especially if you're looking for a pharma-grade product, it has to be extremely pure. So a lot of cost and effort is spent on just pure extracting all these extra molecules that get in the way and could be potentially harmful. And in enzymatic process, again, you don't have that challenge. There are also more kind of nuanced advantages for hyaluronic acid in the enzymatic process, where you can control the exact, so it's a molecule of a certain length, and you want to have a very, very specific length, both for FDA approval. So once you get approved for a certain length, it has to be very, very dependable and constant.
It's really kind of low fault tolerance in that.
Exactly right.
I get the technical similarity with the things, but what are the go-to-market implications? Seems like the different buyers have 3 different potential.
Exactly. So excellent question, and we get that all the time. And that honestly, that's part of the challenges for a platform company. Ideally, I see Enzymit turning into a DuPont or a BASF, so being a giant that can do a lot of different applications. And that's an example of a company doing kind of both ends of the scale. A lot of this will have to be done with partnerships. We don't want to, I mean, when we scale up to production, we'll probably choose one product we want to do ourselves, and the other products will be in licensing deals. But it's just to show you that it can, and really the bottleneck is the enzyme technology. So what I found interesting when I was talking to manufacturers, for example, the early deals, is a lot of these companies have obviously a good understanding on the market and the application. Some of them know fermentation very well. They have zero understanding in enzyme engineering and what the challenges are and what you can do with enzyme engineering. They don't even know what to ask for. So this is the opportunity that Enzymit is stepping into.
I don't think I've asked you this question before, but just looking at this chart, the first 2 is a 3 to 5x win on cost, 5 to 10x. The other one is just less than.
So right now, biojet fuel is an interesting application because right now it's being subsidized.
Yeah.
So the $5 per gallon cost is actually after subsidies, and it's still not a real production cost. So if you just hit that $5 per gallon cost,
must be some way to think about the multiplier that you have on different products. So like early on, I think you and I have chatted about, we talked about kind of synthetic distance from petroleum feedstock. So the more synthetic steps you have, enzyme at each and every of those steps is going to be more efficient. That's a kind of exponential process.
So with biojet fuel, at least the process we're looking at right now is a single step conversion.
All these are single enzyme.
So in biojet fuel, there are with hyaluronic acid and human nucleic saccharides, they're more enzymes, between 4 to 5, depending on the process specifically. But again, the key nut to crack, so to speak, is the durability issue. So if you have a very, very durable enzyme, even at $200 per kg, which I think is a very comfortable estimation. I mean, I've also seen estimations that to go down to $20 per kg of enzyme, but really the number that matters is the durability. So getting in as many cycles out of the enzyme before you have to swap it out. And again, you already have extant examples of cell-free manufacturing for these similar products. So gauzes and phosphors today are produced in a cell-free system, just using maybe 2 enzymes. So I definitely think that's a very, very realistic goal to hit.
And for anybody on the call as well, if you want to drop questions into the chat, we can get to them in for sure.
So maybe I can take the opportunity to also show some results, like real world applications.
Yeah, let's get into some of the things you guys have knocked over already, and some of the wins you guys have had.
So I'm just gonna skip out to my social. Yeah, so sharing. So I'd love to share some success stories that we had with collaborations that we did. So this is an interesting collaboration we did with a cultivated meat company. So as you all know, cultivated meat is a big industry. And for those of you who don't know, essentially what it means is you take cells, trying to be a big industry. Yes. In Israel, I can tell you that in Israel it's a big industry. But so essentially the technology they're using today is you're taking cells from essentially muscle cells from cows or other farm animals, and you're essentially growing them in a vat. But one of the things you need to give the cells in order to mature them is a type of proteins called growth factors. So some of you might have heard about growth factors before. HGH is a common one, but this is a very expensive and prohibitively expensive molecule to add to cell lines, especially if you want to make a commodity. So what this cultivated meat company wanted us, they approached us and said, listen, one of our, as a first target, we're using insulin, we're using bovine insulin to stimulate these cells, but it's very expensive. Can you make a new version of insulin, which is cheaper to produce and more active than what we're using? So by making it more active, you can use smaller amounts and still get the same effect. So what I'm showing you here is a number. So this is a representation of a protein structure. So these are different protein structures that we've actually tested and how they look comparative to bovine insulin. So these are already experimental results. So we've created 50 new designs, and this shows you the expression level. So the way that proteins are currently produced pretty much everywhere is also in fermentation. So you give bacteria the DNA to produce your protein, and you measure the concentration of the protein you get. So this red dashed line here is the amount of the baseline, essentially how much protein you get if you use natural bovine insulin. And our best designs have a 3x fold improvement in expression. So you get 3 times the amount of protein compared to the natural bovine insulin. There's also an advantage here. It's kind of going into a little bit of the technical aspects, but when you express insulin in bacteria, you have to do an additional processing step in order to make that insulin active. For our insulin designs, you don't have to do that extra processing step, so it also saves time and money in that aspect. And when we looked at the activity, we saw that so this is fold improvement over the native insulin that the company was using. We've tested it in 2 concentration doses, high concentration and low concentration. And one of our better designs had a 6-fold improvement in activity compared to the native insulin at low concentrations. And just this morning, I got a call from our collaborators at the cultivated meat industry, and they've actually took our designs, tested them in their own cell lines, and they saw that they could use a markedly low concentration of our insulin and still get the same result. So for the targets they set for us in this project, we blew past their requested targets. So a great way to start the day.
And you mentioned their target was about half.
Right. So their target was just to reduce the concentration they needed by half, and we definitely went below that. We don't know exactly by how much, but it's less than what they wanted in the first place. So a great testament to our capabilities. Another very cool project that we're involved in is designing bacterial sensors for contaminants, and specifically in this case for landmines, for buried landmines. So again, another computational design protein project that we wanted to take also to improve our capabilities. It was also overlapping with another technology we're developing. So essentially what I'm showing you here on the left, so this is a field experiment that was done where landmines were buried in different positions, and you have like this heat map. And once you spray the bacteria, the more intense the color, the stronger the signal. So this is an indication that it can detect something in the ground. So what we did is we designed the protein component, and this illustration will be this black box over here, that once it interacts with the target molecule, it activates a genetic circuit that makes the bacteria fluoresce. And this is results, as you can see, direct results from experiments that we did. So the baseline that they gave us to start with is this blue line down here. And all of these different lines are different designs that we've tested. And to explain what the graph is, so the x-axis is time and the y-axis is signal. So we're looking for two characteristics here. We want to see early response. So as soon as possible, we want to see if there is or isn't something in the ground. And also, of course, intensity is also makes it easier to detect using some sort of, for example, drone. This is actually a plate of the fluorescent bacteria, how we tested these the different designs. And again, overwhelming success, and we're already contracted to expand this platform to detect other types of molecules for future projects.
This might be a very dumb question, but how usable are the enzymes that you make, in the sense that, say, the meat company enzymes producing a different meat company, can they use enzymes, or do they need to go through the same process in terms of figuring out whether
Use, if they wanted to use what we are creating or recreate it? Yeah, it's ubiquitous. If they're looking,
industry uses, so independent of.
You're saying if they nail a growth factor, can other meat companies also use it?
Yeah, absolutely. So it's actually, just to show you the power of this platform of what Enzymit has developed. Food tech in Israel is very big. It's probably the biggest biotech industry right now in Israel. And there are two companies that were independently started just to focus on this problem of growth factor engineering. And we knocked it out in less than 6 months. So we signed a deal with this cultivated meat industry. This is very early on in our lifetime. So it was like, I think we signed a deal less than a year after we started. The deal, of course, includes royalties. So whatever they're using to produce their cells, we get a cut of that. And also, if they end up, like you asked, if they want to sell it to other meat manufacturers, we also get a bigger cut of that.
But are you guys saying how you protect this from others using?
No, I'm actually more in terms of thinking about the economics. Like I said,
how do you sell this to every?
Yeah, because if you have to go through the cost of like, if let's say I have to test the line, so I'm thinking in terms of like trying to cost value. If the time cost value for you basically makes my time cost value zero, then it's awesome. Yeah, but if it doesn't make any difference, then it's kind of scaling. That's really good.
So I just want to take this opportunity to brag a little bit more. If I may. So this is a general bar chart that we created to kind of show our progress over time. So what we're showing you here are two different parameters that we've measured. So every time we've ordered enzyme designs that we've created, or when I say ordered, maybe I'll talk about that for a second. So the process is that we use a computational algorithm to design a protein, but in order to synthesize the protein, you need a DNA that you put into bacteria. So you have companies like Twist and GenScript that synthesize DNA per order for your request. So the computer designs a protein, spits out the DNA sequence. We order the DNA sequence, we get it shipped over to us. We put it in the bacteria, and then we get the bacteria, and then we do the subsequent testing. So essentially, as I said before, there are two very, very important parameters to show that this technology can be cost effective. Number one is the stability of the protein. So one way of measuring the stability of a protein is testing how hot do you need to essentially cook the protein before it breaks down. So essentially it's the same process with cooking an egg. If the proteins in the egg were super heat resistant, you would never be able to cook it on a pan. So typically, natural proteins have a melting temperature around room temperature. I mean, they don't need to be more resistant than that. And that's why, if you boil water, you kill off the bacteria. But so every time we created enzymes, we tested their melting temperature, and you can see there is a continuous improvement in our ability to design. So this is not an iterative improvement. We're not taking the same enzyme and continuously working on it. Every time we tested a new batch of enzymes, this was straight from the computer, no iterative work. So our last batch, we already had enzymes that were near boiling temperature. So the standard deviation is just a spread of the different. The bar plot is a average of the melting temperature, but some of the proteins had a melting temperature of near 100 degrees. And again, I can't emphasize this enough. This is one-shot computational design.
And quick clarifying question there. So you mentioned the stability piece earlier, and this is not just stability at high temperature, but this is also stability that drives the
Exactly.
stability of the enzyme.
Exactly.
Okay, so this is part of how you get 1000x.
Exactly right. Exactly right. So the melting temperature is a very quick way of testing the durability of the enzyme. So obviously most reactions, for example, in sugar reactions, you don't need to run them at 100 degrees. You typically run them at 50 degrees. So the enzymes just have a longer lifespan at lower temperatures. The other really cool, so each of these, by the way, is an algorithm we've developed to specifically test for these parameters. The idea is that the more you can do computationally, the less you have to spend experimentally. So what we've tested on the right side is the amount of protein we get per microliter of bacteria, essentially. So again, you can see here that we see a constant improvement we call this titer, so amount of concentration of protein per microliter of bacteria. These are enzymes.
Marcus, what's typical Novozyme is 10 grams per liter of titers?
Those numbers are not public, so it's proprietary. But the other thing with enzyme production is also time it takes. So it's kind of volumetric, but also how many days do you grow the microbe. So there are fungi that do 100 grams per liter. They grow for more than a week.
But just to put another finer point of that, and this, I don't know if people on Zoom can hear the side conversations, but so Joe asked if there's like what is the industry standard for protein concentrations. My knowledge is anything above 10 grams, you're already.
50 grams per liter.
And this is not optimized. So this is in very, very small scale, not in fermentation, not in a optimized process. So again, when we move forward to scale up our production, we're not going to do our own enzyme manufacturing at scale ourselves. We're just going to go to a fermentation house and say, here's our plasmid, here's our bacteria, grow this at scale and give us back the protein. But so we still have a way of controlling that, but there's also auxiliary technology that exists out there that's also dropping the cost of manufacturing. So, yeah.
Gotcha. I was gonna say we probably should wrap up this piece of it in a few minutes. I wanted to give you a chance if there's other wins you guys have had or other kind of results you wanted to highlight. I also wanted to, you mentioned it earlier in the presentation, but a little bit more detail. I'd be curious on how you guys are breaking that hydrocarbon carbohydrate cycle and how kind of the platform can feed itself over time.
Right. So let's see if I have this slide here. If not, I'll just do the voiceover. But let's see if it's perfect. So so this is a really good question by Teddy. So right now we're here. Enzymit is here. Our current production process uses renewable resources of hydrocarbons, switchgrass, starch, corn, palm oil, using an enzymatic conversion process to turn them into valuable outputs. But ideally, we want to be all the way back here, taking carbon dioxide, hydrogen. Essentially, that's what you need. This is really the underlying connection here. So once you have a carbon attached to hydrogen, now you have a molecule's energy, and you can use that energy to drive synthesis. So we've looked at enzymatic processes for carbon CO2 fixation, but honestly, if somebody else comes in and says, we found a cheap way of taking carbon dioxide and hydrogenating it, it's a win for everybody and a win for us, because really, when we did a bit of a back of the envelope calculation to see if it's realistic. So right now, a cost, for example, of a ton of starch, which is probably one of the cheaper carbon sources, is around $200 per ton. If you go on the, for example, on the Climeworks website, which is a carbon fixation, direct air capture company, they claim they can capture carbon for about $100 per ton. So, and hydrogen is also a very, very cheap material, and it'll keep going down in cost. So if you can use carbon from the air as your feedstock at $100 per ton, there's really no need to go through this step. Essentially, currently what's going on is that nature is going from carbon dioxide to sugars, but technology can probably do it better. So eventually, we're not going to use these feedstocks. We're going to go all the way back here and use hydrogenated carbon dioxide to produce the same materials we're producing now. So yeah, that's a long-term vision, but it's not a pipe dream. I mean, the numbers work out. So yeah, any other questions before we run out of time?
I want to check the chat real quick, see if anybody dropped any questions in there. But just want to remind everyone that stop share here. Just want to remind everyone we will send out a recording for everyone, and we've got a quick question here from Omer, which is, what does the enzyme stability depend upon? Specific protein structure, also dependent on substrate. Where are you introducing the enzyme in the process? Can you talk a little bit about what really drives that?
Sure. So it's a very good question. It's a fundamental science question. So essentially, a protein is a combination of amino acids. You have 20 natural amino acids, and those amino acids, the combination of the amino acids essentially control the properties of the protein. So it's a lot like playing with Legos. You can modify different amino acids and get dramatic changes in the protein parameters. So you can take the same enzyme and make it more pH resistant, more heat resistant, or you might actually want to go the other direction and make it more cold resistant. That's another interesting area to apply this technology towards. So the computational algorithms that we've developed and use are essentially take all this information about amino acids and how they influence proteins and use this information in order to improve, for example, in this case, the stability of the protein.
Makes sense. One last question for me. What are you most excited about next 12, 18 months for you guys, for the industry, but what's on the horizon that's got you excited?
So honestly, for us specifically, there's no shortage of opportunities, and it's really a lot about focusing. So I really want to grow the company to have more programs running in parallel, but also to start scaling up. So for this year, by the end of 2023, we want to show that we have a process working, grand scale products. And once we have that proof point, the next step is to just scale up. So bring engineers to help us go to kilogram scale and then hopefully to ton scale. And for the industry as a whole, I think the consensus is already there. People understand now that bioproduction and the whole biorevolution in general is becoming a reality. There are a lot of kind of surrounding technologies that come together to make this a reality. So you have a lot of computational science advancements that have really helped drive a lot of innovation, but you also have DNA synthesis technology and DNA sequencing technology. So we have more data than ever to help improve our future models. Engineering has also helped push this field forward. So now, I mean, for Enzymit, the fact that we can do so much with really not a lot of funding is because a lot of the cost barriers that existed, 10 years ago have really gone down and are still going down. So it's great news for everybody. We can make products cheaper and more accessible than we could do ever before.
Sweet, I'm excited too. Thank you, Gideon. Thank you, everybody on the line for joining. We'll send out a recording. Keep an eye out for that, but feel free to shoot me or Gideon a note afterward if you want to chat about anything. So thank you, guys.
Thank you.